Hey, hello and welcome back again to another video tutorial of Roy's Academy. In this video tutorial, I'm going to discuss about the chapter 2 of Sir Avery, Macleod and McCarty's experiment, which was performed in 1944 in the middle of 20th century in order to validate the hunt for that transforming material or the biochemical material or that molecule that was able to carry the or convey the information forward from one generation to another or from one cell to another, and that helped in acquiring all the informational characteristics or the functional characteristics innately or as a mean of root cause development for a particular cell. Now in the chapter 1 I've discussed how they performed the experiment. Okay, in the chapter 2 the experimental setup was pretty much similar, but in the chapter 2 there were some modifications made in order to have the stronghold or in order to verify the inert in order to verify the inert fact or the inert phenomena that DNA was the only genetic material which helped in the transformation principle as per the legacy of Sir Frederick Griffith of 1928's experiment. And the DNA was that only biochemical material, or the nature of the biochemical material was genetic material or DNA, that is nucleotide, which helped in incorporating the specific functionalities or the characteristic feature, and that led to the development of certain or this led to the development of to the entire organism, as along with all other characteristic traits or features in the body of that organism, starting from the development of a single cell to the multicellular or the multiplex system of the tissue and organ system, and each and every functionalities of a particular organism's body was being manifested or comprehended by the DNA only. So let's see in chapter 2 what they did exactly. What was the difference between the chapter 1 and 2? It would be clear in course of my discussion. Now in chapter 2 they did they took the strain of the bacteria was similar. The strain of the bacteria was nothing but the Streptococcus strain only. Okay. In chapter 2, the Streptococcus pneumonia was the only strain. And now in this chapter, again they considered few other case studies. Okay. They considered few other case studies, like in chapter 1 they did. Let me write down the case studies first. So, case 1 again. Okay. Then case 2 again, case 3 again, and case 4 again. Now as I mentioned earlier that the R2 type strain was non-virulent, which was not pneumonia causing or not lethal at all, and the S3 type strain was pneumonia causing strain of the Streptococcus or the Diplococcus pneumonia. Now in this particular chapter 2 of the phase of the experiment they conducted, in this particular chapter 2 what they did they arranged the experimental setup in a bit different manner. Okay, in the first setup, let me draw all the setup tubes first, or the culture tubes, which comprehended or comprised the four cases of chapter 2. Okay, in this chapter 2, in case 1, what they did, they took the DNA extract, the DNA extract of the S3 type bacteria. Okay, the DNA of S3 plus living R2 type bacteria. Okay, In this chapter 2 they did this in the first case. In the second case of the setup of experiment, they took the protein, they isolated, they took the DNA only, the DNA of the S3 type, DNA of S3 plus living R2 type bacteria, and they supplied this second culture tube with an enzyme known as protease. Okay. Now in the third phase, in the third phase also they did the same thing. They took the DNA of S3 plus living R2 and R2 type bacteria and supplied it with RNase. RNase is actually the RNA denaturating enzyme or RNA destroying enzyme. And in the fourth case, they took the DNA of S3 plus the living R2 type and supplied it with DNase. DNase was the enzyme which helped in denaturing the DNA. Now, they did a particular modification, a major modification was done by them in this particular setup, entire setup of experiment. I am telling you what the modification they did. Okay. They heat killed the DNA of S3 type before supplying it with the culture of R2 type, living R2 type. Okay. In all the cases, the DNA was heat killed. Here also it was heat killed. Here also the same phenomena was done. It was heat killed, and here also it was heat killed. Okay. Now what they did? In the course of observation, when they incubated this entire 4 setup, okay, they incubated the entire 4 setup and transferred them to the Petri plate. They transferred them to the Petri plate. Okay. And in all these setups, when they transferred this culture to the Petri plate, the heat killed DNA of S3 plus the living R2 type, the heat killed DNA of S3 plus living R2 type plus protease, the heat killed DNA of S3 plus living R2 type plus RNase, and the heat killed DNA of the S3 type plus living R2 type plus DNase. Okay. Now in the first setup, what happened? The living R2 type was present. So in that case, the heat killed DNA of S3 was supplied right in the culture medium, and the incubation was done for subsequent specific period of time. But in the observation period, in the Petri plate, the S3 was found. The S3 colonies were found. Okay, in the observation period. Now, in the second setup also, in the same way, in the second setup, what they found, that heat killed DNA of S3 was given along with the living R2 type plus protease. Now, protease is a protein denaturating enzyme. It destroys the protein, okay, out from the medium. But still, in this case also, the S3 type colonies developed. The S3 type colonies were only found. And in the third case, what happened? The RNAs were supplied. RNAs denatures the RNA, like I said. In this case also, S3 colonies were found. S3 was found in this case also. But in the fourth case, something strange happened. In the fourth case, what happened? DNase was given. DNase destroyed the DNA, heat-killed DNA of S3 type, whereas the living R2 type strain was entirely, the complete viral particle, bacterial particle was present, the bacterial, the mature bacteroid was present, right? The living R2 type, the mature bacteria, entire bacterial cell or the complete cell of the bacteroid was present, and the DNA of S3 was heat-killed and given. Now DNA denatures the DNA. In this case, what happens? No S3 colonies developed. Okay. No S3 colonies developed at all. Only some living R2 type strains were found. So, what happened in this chapter 2? They modified the chapter 2 by supplying the heat-killed S3 type DNA, and unlike chapter 1, in chapter 1 what they did, they supplied the DNA without heat killing. In this chapter 2 they heat killed the DNA, and in chapter 2 the enzymes were changed except DNase. The other two enzymes they added is protease and RNase, and in chapter 1 it was like carbohydrate and protein they isolated and supplied with living R2. Now in this setup what happened, the heat killed S3 type plus the living R2 type, the heat killed S3 type DNA was so dominant and virulent that it led the transformation of the R2 type to the S3. So S3 type colonies were found. And in this setup also, where protease was present, protease was not having any effect on the culture medium of the living R2 and the heat killed DNA of S3 type, then also the S3 colonies developed. And in this fourth case, RNase was not having any effect on the DNA of the S3 and the living R2 type entire bacteroid. So this case S3 also developed. And in the fourth case, the DNase denatured or destroyed the DNA of S3 type. So no S3 colonies were found, only the R2 colonies developed. Now so what happened but basically here? In this case, what happened? Let me recall the experiment performed by Sir Griffith in 1928. Sir Griffith was unaware of the biochemical nature of the genetic material or the exact molecule present in the cell or the biomolecule that or that element that was having that inert capability to perform this specific inheritable characteristic or transformation characteristic, that the genetic material altered itself and started performing in a different course of manner, in a different type of genetic element. Now the same thing they put forward with in the chapter 2 Avery, Macleod and McCarty with more detailed and more non-speculated, more firm belief or validated fact with explicit organization of their experimental setup, with more potent or validated, with more modified, robust and rational sense of their experimentation. They modified the experiment and brought the robust phenomena in limelight, that the heat-killed S3 type DNA finally led the transformation of the non-virulent living R2 type to the S3 colonies. And in Griffith's experiment also, the heat-killed S3 type was given. In that case also the same phenomena was happening. The living R2 type converted into the living S3 type. And in this case, when the heat-killed DNA was given, then also the living R2 type converted to the S3 type living colonies. In the second case, in the presence of protease, the same phenomena happened. In the third case, in presence of DNase, the same phenomena was happening. So it was more robust and rational in sense and very radical approach by what was performed by Avery, Macleod and McCarty in order to put forward into the limelight or into the broad spectrum the legacy of Sir Frederick Griffith, the hunt for the genetic material or the hunt for the biomolecule or the biochemical element that led to the characteristic development of a particular species or a particular cell along with its all functional A to Z functionalities. So in the fourth case, when DNase was given, DNase destroyed or removed the DNA of S3 type, and the living R2 type was present, though so only the R2 type strain survived. In this case, as the DNA, though it was heat killed, was not present in the cultured medium at all, then also the R2 type only developed, and since the DNA was totally abolished from the medium, so no S3 colonies developed. So you can understand that how robust or radical approach it was, or very logical or smart amelioration or improvement of the legacy of Sir Frederick Griffith was done in order to validate the same phenomena, that DNA acted as that biochemical element which led to the conversion of all this, led to the observations of all these facts. Because without the DNA, the development of the cell starting from its DNA was the root cause of everything for the incorporation or acquisition of any inheritable characteristics and conveying that particular information from one to another generation, successive generations. Okay, so this led to the final conclusion that DNA stood to be that biochemical element or the biomolecule which stood to be that particular medium for conveying the message or the imprinting the message of inheriting a particular trait or as function from one cell to another or from one generation to another, provided that it can also incorporate some alternate characteristics and can change its functionalities or change its nature into that particular type, into that alternate or new nature, or acquire some new natures from another cell and can develop into that particular cell or that particular DNA of other organism which the DNA was not having previously. Okay, so this is what all about how Sir Avery, Macleod and McCarty performed their experiment and brought forward in limelight of the entire legacy of the DNA as the transforming material. Okay, the same principle is nothing but transformation, which Sir Frederick Griffith did. The DNA got transformed. The test for this genetic material led to the phenomena of the transformation of the DNA. The DNA helps in acquiring the foreign genetic element into itself and thereby develops itself into a newer kind of genetic element. Okay, and perform new functions. So if you like this video, share the video as much as possible, hit the like button and comment down in the comment section below, and also subscribe this channel positively in order to get such more updates. Thank you.